Welcome to the podcast, Cheryl.
Thank you, Lindsay. I've been so excited to come on your show. You know why?
Why is that?
Before I started Tiny Health, and this was maybe even before 2020, I think I just kind of searched on Shopify, or I keep saying Shopify, Spotify. What are all the stool or gut health podcasts out there? And I found yours.
Mhm.
And so I bookmarked your podcast. It's been there for years, and yeah, so it's such an honor to finally come on board to your show, because this is literally before I started Tiny Health when I found you.
Awesome. Well, I'm glad to have you here. So what led you to start Tiny Health?
In 2020, I gave birth to my son, and that's the year I also started Tiny Health. Well, I started the company a week after he was born.
It's ambitious.
Well, yeah, I incorporated the company, and I self-funded a study with 8 moms who was pregnant around the same time I was, and giving birth the same time I was. So technically, I started the study before I started Tiny Health, in that, my story goes back even before to my daughter. 2 years before my son was born. So in 2018, I gave birth to my daughter by a C-section. And I was like really researching, the impact of C-section to my child. At the time, I didn't know the linkage between C-section and vaginal birth versus gut health. But then since then, I've uncovered that, like your gut health, your gut microbiome, where do you get your gut microbes from? And the answer is from your mom at birth. You first get some vaginal microbes from labor and going through the vaginal canal, and then some gut microbes from the fecal fluid during labor. And then breastfeeding, through breastfeeding, the mom is continuing to transfer some gut microbes from her gut to baby's gut. So then I was uncovering all this research about how not bypassing the vaginal canal can lead to the baby having higher risk of eczema, allergies, and asthma. And so I was kind of alarmed, obviously, for the C-section I couldn't control, and also trying to figure out if I can rebalance my C-section baby's gut and restore her gut, the missing bacteria that wasn't there, and to prevent her risk of chronic conditions. But obviously, I was in that research mode, so I didn't I didn't get to see her gut. So when I was pregnant with my son, I was like, this time, if I had to have a second C-section, I really wanted to know sooner than later to course correct earlier on, because my daughter did end up getting eczema around 6 months, food sensitivities, she's dairy, gluten intolerant, so she has a lot of gut issues and skin issues, and really wanted to prevent that for my daughter.
Yeah, my son was also C-section, and I did give him some probiotics sometime, I think, but it may have been later on. But I was certainly aware of the issue at that point, or at
Mhm.
least maybe a year or two after, I think.
Mhm. Mhm.
So why do you recommend that expectant mothers test their microbiome?
Oh, yes. So, we started Tiny Health. So then to kind of follow on with my story, and I'll come back to your question,
Yeah.
I self-funded a study with 9 moms, including myself, and tested their microbiome throughout the first year of life. And then once I learned that I can establish that, and I, sort of gathered scientists and microbiologists who knew much more about this than I did to help me build this thing. And then we spent another like 2 years of R&D before launching the tests in 2022. And our flagship product was the pregnancy sort of gut test and vaginal tests, and then also the newborn gut tests, or basically in the first 3 years, which was a huge gap in the market. There was no stool tests meant for babies and moms at the time. And so with my son, I mentioned earlier, then I had that ability to test him, and myself too. I was like, with the study that I funded, I realized through my pregnancy microbiome that I didn't have the BIVs or the beneficial bacteria that I was supposed to transfer to my son through a vaginal birth and through breastfeeding. So if mom's gut was deficient, then even if she had a vaginal birth and she was breastfeeding, she's not really transferring those microbes. Dr. Martin Blazier is a really well-known scientist or microbiologist.
I know
You've
him
heard
well.
of him. Yeah,
Yeah.
he wrote this book called Missing
Missing
Microbes,
microbes.
right?
Yep. That was what
Exactly.
launched my interest in the gut microbiome.
Amazing. Yes. And yeah, he's well respected. And I think there's even a documentary now that he created with his wife. And so he talks about how overuse of antibiotics is really causing a depletion of these essential microbes in our guts, right? And so that was the microbe that was depleted in my gut. I didn't have any bifidobacteria. I had
Mhm.
zero. And that is the most essential microbe that a mom should have in a way to transfer to her newborn, her baby at birth, either through labor and birth or through breast milk. So it's interesting now that we've been in the market for about 2 years. We've seen over 25,000 families. So we have a lot of samples, a lot of infant samples too. In 30% of the cases where the child was vaginally born and breastfed, they didn't have any BIVs. And where we had mom's gut sample, the mom didn't have any BIVs either. So it's really interesting. So I would recommend, if I knew what I know now, and if I were to have a third child, not to say I am, I would, try to restore that in my gut before trying to conceive, because it takes months for an adult gut to change.
Mhm.
Like if you and I were deficient in it, it could take us at least 3 to 6 months, sometimes 9 months, to see those BIFs recolonize, because we're trying to make something that wasn't there, maybe that was being destroyed by antibiotics. And that was what I learned. I had to go through this path of like, why am I not, I don't have any BIVs. I asked my mom like how I grew up, and she said I was formula fed 1 month on, and I had antibiotic exposure as a kid. And BIVs are very sensitive to antibiotics in early life, especially in early life. Sometimes it gets wiped out, and if you don't take care to restore it, it
Mhm.
may be gone forever, right? And so that's kind of why I think like everyone should do a stool test to really just see if there's any BIVs or Archimencia, and Archimencia is the other crucial bacteria that are important for your metabolic health and gut lining. I would check in if you're trying to conceive or pregnant, if you have BIVs and Archimencia in your
Mhm.
gut, because these are two quite important bacteria to pass on to your child.
And is it, if you're gonna reestablish it, is it Bifidobacterium infantis in particular that you wanna restore? Or is any bifido or just a combo bifido? What do you guys recommend in that scenario?
Yeah, that's great. A great question. So it depends, right? If you're an adult with no BIVs, frankly, any BIVs could be beneficial. But if you are planning to have a child, you really are looking for 4 specific BIVs, which is what you mentioned, infantis. B infantis is probably the most best adapted at digesting mom's breast milk, or specifically the HMOs in mom's breast milk, human milk oligosaccharides. HMOs are the prebiotic fibers that makes up a third of mom's breast milk that the baby cannot digest. It is there for the bacteria, the BIVs of the baby's in the baby's gut to digest. And so B infantis is the best adapted in digesting the HMOs in mom's breast milk. The second best, when maybe in no particular order, the next best are B bifidum, B longum, and B breve. So we really want to see these 4 specific strains that are very good at digesting mom's breast milk for the best child health. In the first year, we want to see these at 50 to 90% of the infant's gut, frankly. And then as they grow older, they get more exposed to solids, nature, pets, their guts diversify, and you don't want to ever see 90% BIVs in a toddler's gut at 2 years old. So what I was researching and reading up the research, the first 6 months of life, the first year, there's a very specific trajectory of gut maturation that an infant should be following. They're best adapted at training their immune system, because the baby doesn't have any gut microbiome in the womb. There was some controversy back then and debate about, oh, maybe there is, there isn't, but the science is pretty settled there in the recent year, that the major transfer, the colonization happens at birth. There's some passing through, but it's not really, there's no colonization in the womb.
Mhm.
So what this means is like baby is born pretty much without an immune system, and their immune system is being trained by these bacteria, these microbes, right? So the initial kind of like, I always describe the gut as a theater room or like, yeah, like a theater room with only like say 10 seats at birth, right? And as the infant grows, the seats in the theater expands. When they start solids, it may expand to 20 at first when they're just kind of nibbling stuff. And then when their solids have been established, it may expand to 50. And then as they grow up past 1 year, it's quickly expanding to 100 seats, 200. You and I now at our age probably have 300 to 500 species, meaning 300 to 500 seats in that theater. So our theater is huge and very complex by now, but an infant's theater is very small and developing. So that's why those early years are so crucial to get the right balance, because this right balance of beneficial versus unfriendly bacteria is there to train their immune system, what's friend, what's foe, to recognize if they should react to it. So the reason why eczema, allergies, and asthma is so tied to like the gut microbiome in the early days, and there's a sign of dysbiosis linking to these, is because like it's a sign that their immune system wasn't trained correctly by the right balance of bugs.
It's
If
overreactive.
it's overreactive, yeah, it's pro-inflammatory. In these babies' guts, we often see zero bifidobacteria. So instead of the BIVs that we just mentioned, the 4 BIVs, taking 9 out of 10 seats in the theater, it's the other way. It's maybe 0, and then 9 out of 10 are E. coli type bugs, Klebsiella, maybe Streptococcus, Staphylococcus, very
All
pro-inflammatory.
opportunistic sort of bacteria that at higher levels are pathogenic.
Mm-hmm. And
Yeah.
babies can tolerate that much. Sometimes parents are shocked, like 90% E. coli, is my baby sick? And the answer is no. Babies, again, are born without an immune system, and they can tolerate really high amounts of unfriendly bacteria. But you do see it in terms of symptoms. You get a colicky baby, a gassy baby, a baby who can't sleep very well. And then you get eczema when they start solids at 6 months. So it's kind of like that high level of unfriendly bacteria, of opportunistic pathogens, is sitting there creating this really inflammatory event in their guts and not training their immune system properly. And so when they finally are met with a dietary allergen or dietary, yeah, like, new thing at 6 months, they get triggered and then the eczema happens.
Yeah.
Um,
And so, given that you guys now have sequenced all these infant microbiomes, I'm curious because, there are different strains of Bifido infantis, and some are a lot more expensive than others.
mhm.
So I'm wondering, is the Bifido infantis you're finding in the guts of healthy babies who have the right microbiome the same as any one of those strains they're selling out there as probiotics? Or are there a number of different possible Bifido infantises that are good enough?
Yeah, this is a great question. B. infantis, we've seen some native B. infantises from mom and baby, and it's really hard to say. We would have to do a proper study, and we do have the data now to really dive into it. I have to admit, we haven't done that yet, but this is what we could do in the future, right? In terms of like, is the innate strain better than a supplemented probiotic? Who knows? Probably, right? If the mom had the infantis, it's likely, like hard to say superior, because her breast milk and her HMOs in the breast milk is probably adapted somewhat to her own B. infantis and to help it colonize in the infant's gut. But again, the problem with our society and modern lifestyle is that most adults are missing BIFs, including myself, right? And a lot of population it's missing. So then when that happens, the infant really needs a supplemented B. infantis that is well studied, clinically backed, and they're not all equal. When you talk about probiotics, you can't just grab a bottle from the shelf and think, oh yeah, I'm taking probiotics, or I'm giving my child a probiotic and I'll be fine. No, because different strains have different functions. And even within the strain, like you mentioned, there's so many different strains. And when for your audience, when I talk about strain, they are the numbers and letters behind the probiotic. So for example, EBC, like B. infantis EBC001 is one that we recommend. The brand name is Avivo.
Right.
And it is the most clinically backed B. infantis for infants. but even I would do it for adults too, because it's so potent. And if an adult is missing some bifs, I would take that to help it recolonize in your gut by
I actually
adding it.
just bought some, and I'm gonna make the Bifido yogurt
Nice.
with it so I can make it
Now,
expand.
did you buy the HMO supplement along with it?
I did not buy any HMOs yet, no.
Okay. If you wanted to recolonize, you have to have the food, the prebiotic or the probiotic to colonize. Otherwise, you have to keep taking the probiotic, right? Because it is going to give you a transient effect. But again, without the food there, they're not going to stick around. They're just going to pass through and give you that benefit, and you have to keep taking it every day.
The
But
rest of my life.
ideally, exactly. But ideally, the goal is to help it recolonize in your gut, by, again, I think it took me 3 months of pre and probiotic combination. And then I sustained it with dietary, good diet that supports the sustenance of these probiotics once they've colonized in your gut, right? High fiber diet, polyphenol rich foods, XYZ. So it takes effort. So in an adult, right, it does take, which is why coming back to the mom question, like before, if you do want to conceive in a year, I would start now and start restoring your gut now, because it may take you 6 months to get there. And then hopefully once you help it recolonize, it's like I had 0 and I managed to get it up to 10, and then it became 8% for a long time without any supplementation. And now it's kind of back down to 0 because I moved to Texas from California, and I realized my water supply here is terrible. We tested our water, and there's arsenic and lead and uranium in my water. That's a different story. But it goes to show you how many factors externally, environmentally, and dietary wise could influence your gut, which is kind of why we encourage proactive testing twice a year, just, as an adult or an older child, so that you catch these things before they they turn into symptoms.
Yeah.
So, now I'm trying to work it back up to 8% where I was.
Yeah. I mean, I wonder whether you really can recolonize, whether you do have to essentially supplement with the probiotic supplement, or take the prebiotic once you supplement with the probiotic indefinitely in order to keep these strains alive if they haven't naturally settled. Because I know the gut microbiome of an adult is a lot more settled than that of a child. Like, it's easier to
Mhm.
alter it in a child. So I mean, normally, what would you say are the primary differences then between like the infant microbiome and then, say, when does it start to settle into the adult microbiome?
Yeah, that's a great question. So if you go back to the theater example of it being 10 seats at birth and then it increasing over time as they're licking the floor, and if you have a pet, even better, then the pets are bringing in that diversity from outdoors into indoors, which is why there are studies showing that if you have a dog or a cat at home, your kids are less likely to have food allergies, because it's bringing in a lot of diversity that we're missing in a modern lifestyle, right? So kind of like, that, very like that, the infant gut is malleable in the early years because it's so small, and it's easier if you think about it. Oh, you just put in like 9, good guys and 1, bad guy inhabiting the theater room, or let's just say the flip side, right? 9 bad guys and 1 good guy happen to be in that theater room in the first few months of life. It's easier for you to come in and be like, hey, move away, let me give you a specific strain of probiotic, or breastfeed. If mom had the right bacteria and mom breastfeed, that is very healing. If mom had B. infantis, you just have to breastfeed. You don't even need to supplement with a probiotic. And mom's B. infantis can come in and chase those bad guys out and take those seeds. So it's very easy in an infant gut, frankly, as long as breastfeeding is involved because of the HMO. So the key here is the HMO in a mom's breast milk is the modifier, really, of a dysbiotic gut or a very imbalanced gut. So that gut changes over time, and with solids and all that. Again, like there are some infant guts who have then too much beneficial and no unfriendly, 0 E. coli, and that's not healthy either, because we need some unfriendlies to train the immune system. Like 1, out of 10 should be unfriendly. So, in literature, this is kind of what we're seeing, and even in like clinically, because we do a lot of survey data on babies, so we have a lot of data on what kind of, makes a healthy infant gut and what isn't, because it's so simple. Like it's so much easier to characterize what is a healthy infant gut and what is an unhealthy infant gut versus for adults, it's much harder to categorize that definitively. And we're not anyway looking for one universal healthy microbiome in adults because there's just, everyone has their unique microbiome. But in infants around the world, if you think about it, in the first 6 months of life, their only food is milk, right? So their gut isn't really influenced by different cultural diets and maybe weather or environments yet. It really is consistent. So in the first 6 months, it's very clear what should be there and what shouldn't be there. And as they eat solids, that's where it gets a bit more complex. I think post 1 year, it gets more complex depending on diet and culture and, all kinds of things, but still malleable because it's still kind of like maybe some kids are still being breastfed at 1 year old. And again, the more there's breastfeeding, the more malleable it is, the easier it is to change the infant gut if there are imbalances. 2 years in a lot in America, most people are not breastfeeding anymore. I did breastfeed my son for 2.5 years,
Wow.
and my daughter for 18 months.
Mm.
So yeah, it's kind of like, obviously everyone has their own personal journeys, but around 3 years of age is when the child's gut reaches what we call adult-like maturation. Or
Mm.
in the literature, it's kind of there's a transition phase between 3 to 5 years old. So I would say maybe a 3-year-old's gut is pretty close to adult-like maturity. 5-year-old is a bit more definitive,
Mhm.
where if we take a 5-year-old a 5-year-old's gut test, and if we have the mom or the dad's stool test, it often maps very similarly. I can tell they're living together, they're in the same family, because the child's microbiome tend to look more like the adult microbiome
Mhm.
by 5.
Do you think that's because they're sharing saliva essentially, or because they're eating the same foods?
All of the above, 100%. And there has been studies lately being published that the family microbiome is a thing, like I can tell, because I've seen thousands, tens and thousands of microbiome results now. If I can tell if a partner, like not husband and a wife, but boyfriend, girlfriend, are they living together or are they not? Because sometimes they do like partner and come in for a consult call and I'm like, you're living together, aren't you? Or you're not living together. Or the partner's like, the husband's like working elsewhere and eating a different food. So the living environment, the food, the diet, like everything
Mm.
influences your gut, right? So the more similar things you do as a family, the more similar your microbiomes will be, which is why we always tell parents, like, because, we started with the infant gut and mom, because, babies cry and they have like symptoms and parents are trying to find a way to help to get answers that maybe their pediatricians can't give them, because the gut, the gut of the infant is, there's a lot of evidence in it, but it is relatively new. It's not really in medical school yet, right? It takes an average of 10 to 15 years for groundbreaking academic research to get into medical practice. And for me, I couldn't wait that long. So I really wanted to start this company to bridge that gap. And so for, like parents coming to us, I'm kind of forgetting my point now, actually. What was I? Apologize for that. I think I was talking about, and you can edit this out, right?
I can edit it out of the podcast, not of the
Okay,
video.
sorry. Let me pause and do you
That's
remember
fine.
what we were talking about? I lost my train of thought.
Um, the stabilization of the infant microbiome and getting it closer to the adults and the transition period, and then
I was
came
trying
on
to
from
make a
there.
point and I lost my train of thought. It's okay.
Oh, people living together and what their microbiomes would look like
Oh,
versus living
yes.
apart.
Yes, thank you. So yeah, and in kids, I think we always tell parents, like, they come to us because they are trying to course correct their infant's gut or like rebalance it and make it better. But we're like, what about you? Your child's gut is eventually going to look like yours. So, doing a stool test is, especially if you, from a perspective of your baby's lifelong health as it relates to your immune system training. You can't just course correct in a point of time because this is a lifestyle. It's a repeated thing. You have to have good dietary habits as well. And not everyone needs supplements, but if you do need them, you should know what specifically you need based on your test. Whether you do a stool test for your gut health or a blood test or nutrition test. We always believe in testing and not guessing. And
Yeah.
then lifestyle changes too, right? Like a lot of parents were educating on very toxic household cleaning supplies, antibacterial soap. You know, a lot of parents are now from the pandemic, like the word germs and bacteria and viruses is scary to them. And so we're, overusing these antibacterial soap that gets into our child's mouths and guts, right? And it's not just killing the bad bacteria, it's killing the good bacteria as well. So we're giving a lot of education on lifestyle changes as well. Like people are not spending enough time in nature and outdoors and all that. And it may sound generic, but it is true. Like we are deficient in diversity, and we used to spend 90% of our time outdoors and 10% indoors, but now it's the reverse, right? We're spending 90% indoors and 10% outdoors. So a simple thing like open your window once a day for at least 30 minutes is going to change the microbiome of your environment and your household to invite some of that outside microbiome in to help your child diversify. So all this is to say, like you have to think about your family's microbiome holistically and not just kind of like help your child, but not care about your own microbiome.
Got it. And so you mentioned eczema and asthma and things like that. Are there any other signs and colic, any other signs you might see to think, oh, my child's microbiome might not be properly developing? Or even if like, I know if you had a C-section, that's sort of an obvious likely thing, but if you didn't have that.
Mm-hmm. I mean, because the immune system, the lifelong immune system training of the child is so tied to their gut bacteria at birth, knowing what I know now, again, why wouldn't you test early to get that snapshot of what was there at birth? So the earliest sample you can take is the 7-day sample post-birth, and that will give you a clear idea of what seeded the baby's gut at birth. And then if you wait till maybe 2 to 3 months, you'd start to see if mom was breastfeeding that the breast milk, if mom had the B infantis or bifidobacteria to be transferring to baby's gut. So by then, if baby had 0 bifidobacteria by 3 months, then we know probably the mom didn't have it. And by the way, if dads had it or older siblings have it, they can pass it on to baby too. So sometimes we do see no bifs in mom, no, but and then the infant has bifs, but like where is this from? And when we have an older sibling who maybe went to daycare and had that, and had that colonized from daycare friends or a dad, we see that passing on to infant. So it's really interesting. Like if the baby is completely deficient of it, that means nobody in the household likely has it, and everyone should be working on that thing as well. So yeah, like I think that is quite crucial, and like C-section is definitely a thing because the baby's being exposed to the antibiotics from the procedure and they're not passing the birth canal. However, again, like the breastfeeding is very modified, like the number one modifier of an infant's gut.
Yeah.
So yeah, that is very healing. But there's various things that you can do to restore an infant's gut.
Yeah. But are there any other conditions that you might see in your child that would clue you into the fact, like, say, I'm sure there's people listening who have 1-year-olds and 2-year-olds and 3-year-olds. Like, what might they be seeing in their child that would
Yeah.
let them think, maybe something went wrong with the microbiome?
I mean, the common things in infants, there is a colicky signature or gassiness. Those are very common symptoms. If a baby is crying a lot and maybe not meeting that colic definition specifically, but as a mom, as a dad, you have your gut feeling that something is off, right?
Mhm.
If they're constipated. But by far the most common issues that parents come to us is eczema and allergies.
Mhm.
And now there's a lot of cow's milk protein allergy, kind of symptoms. But again, colic and sleep issues are also very correlated with
Mhm.
infant gut. So almost all of the kind of like common baby symptoms that you can think of
Yeah.
can be linked back to the gut, and it
Okay.
can be linked back to bifidobacteria. And
Now,
for older kids,
having had a child, before you go on, having had a child, I would say I know that loose stool is a normal thing for a breastfed baby, like
correct.
liquidy sort of yellowish stool, right? Is that what it,
Yes.
I my son was breastfed, but also bottle fed because I didn't have enough milk. So I know that they tend to have more solid stool if they're being bottle fed too.
Hmm.
Yeah,
Hmm.
just wanted to bring that up. Now go ahead with what were you going to say with the older
Oh
kids?
yeah, no, like the range of normal is wide in infancy, right? Like, I get asked a lot, like, what if they only poo once a week, but they're breastfed, and that could be normal, or they poo 5 times a day, breastfed, and that's normal, or like formula fed. It's really hard to say, but I think like some stool we're hearing with connected to cow's milk protein allergies, like the mucousy type stool, the green stool, again, like your gut tells you something is off, you kind of know about it. And I would say just check in and do a stool test. As the baby gets older and are eating solids, we get more constipation issues because then a lot of kids, toddlers don't eat fiber, right? And so, we tend to see like fiber digestion or, maybe low short chain fatty acids in those toddlers. And so that's when we offer some strategies to parents in sneaking kind of like specific fibers in smoothies or, like more in their diet, or encourage their kids to eat more fiber in that sense. So yeah, constipation is a very common thing with older kids. And then, we also see teens who have acne problems, right? Skin issues. So again, there's something called, you've probably heard of the gut-brain axes. There's something called the gut-skin axes, which is similar to kind of like the eczema, acne, skin issues kind of thing. There is an association where, probably there's too much mucus degrading bacteria in the gut that's kind of like running down the gut lining, and that's kind of affecting the skin as well.
Mm. So it's creating some leaky gut for the kids
Yeah, leaky skin, I would say,
and then leaky skin, right? Right.
Yeah, exactly.
Okay.
So there is a connection there, and which is why, like 80% of our immune system is in our gut, and that's why it is very essential to take care of it. And when we're eating, we're feeding the microbes in our gut,
Mhm.
as much as we're feeding ourselves, right?
Yeah.
Yeah.
So have you seen now that you've seen all these tests that all of the kids born via C-section have a perturbed microbiome, or do some of them, or some of them are okay?
No, yeah, it's interesting. So like I mentioned earlier, 70%, sorry, 30% of vaginally born breastfed babies don't have a good gut, right? And I think something like that amount, 30% of C-section born babies have a really good gut.
Okay. So fewer,
And
but some.
yeah, fewer. But again, I think the modifying factor is the breast milk,
Right.
the length of breast milk and whether mom had the bibs to transfer to baby. And
Yeah.
if not, it's okay. It's not the end of the world. Again, this is why we created Tiny Health, to be empowering to parents and not like there's no guilt tripping. It's nobody's fault. Like my mom couldn't breastfeed me, and because
It's
of
hard.
her low supply too, it's hard, right? So
It's hard.
like,
Like you really have to be dead set and determined on it and willing to suffer all sorts of things. And even still, sometimes you might not succeed. I've discovered.
yeah. And it's also, our horrible maternal leave policy in America.
Yeah,
It's
yeah.
also sometimes it's a choice, and that's fine. But we offer ways to support the baby's gut if you're formula feeding and if you had a C-section. There are certain formulas that are better adapted at providing support to the gut, the infant gut, which really it comes down to prebiotics. So I would look for HMO, formula added with the HMOs to again help the bibs colonize. So it just means that the C-section gut may need some more support, and there are means to do it, right, with a lot of the supplements in the market right now. So again,
Yeah.
we help, and sometimes we help validate, because we had a C-section mom cry when we were on a consult call with her because her baby's gut looked perfect, and she was like, I did all this work, and this has validated her work. and we told her to stop the probiotic she was giving the infant because she was giving the wrong kind of probiotic. She was giving a lactobacillus-based probiotic, which didn't change or impact her infant's gut. And so she got rid of it. It wasn't doing anything anyway. And so for her, it was very, very validating as well.
Mhm. So back in the day when, what, maybe 5, 10 years after Martin Blazer's book came out, there was a lot of talk about the fact that babies get the microbiome from the vaginal canal, and that you should reseed your baby's microbiome by taking swabs. And then it all came out that most of it's actually coming from the fecal matter that they're encountering as they're born. So I haven't heard as much about the seeding your baby's microbiome by putting fecal matter in their mouth. I'm just curious what's out there in the world now about that topic.
Yeah. So, Dr. Martin Blazer's wife is Dr. Maria Gloria Dominguez, who is also a well-known researcher in the space in Rutgers University. And she's one of the papers I read when I was researching this. She
Mhm.
was the one who came up with the idea of vaginal seeding after a C-section. So at the time, it was a little bit controversial. In 2018, I would say that was when I had my daughter. I did it anyway, so I did the vaginal seeding before it was sort of like approved. It's still not approved by ACOG yet, to be frank, but there has since been a few more papers coming out to show that the babies swap with the gauze that was put in the mom's vagina for an hour before the C-section operations, and then within 2 minutes after C-section, you would kind of put the gauze in the baby's mouth and face to mimic the kind of like passing of the vaginal canal. The few studies that came out in the recent years did show that those babies look more like vaginally born babies than C-section babies. So I would say there is some proof that it works. And
But they're not taking
but
fecal materials.
no, they're not.
Okay.
And there's also I can explain more here too, what I think the theory is. But I do want to address that. Like now that we have a vaginal test, I would encourage moms to if you're pregnant or trying to conceive, also take a vaginal test. So that if you do have, your plan B, because you have your perfect plan A, which is vaginal birth, and then you always have to have a plan B, right? You shouldn't not have a plan B. I would see if I'm a good candidate for the vaginal swab. So if my vaginal test came back all good, I have high lactobacillus in my vagina canal, and I have no weird bugs that I wouldn't want to swap my baby with, then you would have that option, hopefully with your midwife or your OBGYN to do that. So I would highly encourage that vaginal test also, because if your vaginal microbiome is not in a healthy state, there is risk for preterm labor and other pregnancy, complications. So get that checked. And then as far as some studies showing that mostly babies are colonized with the mom's gut bacteria, it is true. However, our scientists at Tiny Health and even within the scientific community, we have a theory that there is a role of the vaginal microbiome. So lactobacillus is what should be dominating the mom's vaginal canal. It should be 98% lactobacillus and very low diversity. An unhealthy vaginal microbiome is one that's very low lactobacillus, maybe 10% or maybe none. And again, that's linked to high risk of preterm labor and things like that, STIs, STDs. So we think that the baby passing through the vaginal canal, if we get an earlier sample, maybe a 4-day sample, sometimes we don't get it because it's meconium and we don't get bacteria yet. But in some 7-day samples, we do see lactobacillus strains colonizing baby's gut, but it disappears very quickly. It's almost like the lactobacillus are there to prime the baby's gut and prepare for the seeding of BIVs from the mom's gut. So this is the theory, lactobacillus and bifidobacteria are two very commonly common probiotics that you'll find in bottles in the supermarket, right? And the pharmacy and things like that. And we think they go hand in hand. They're kind of they help each other. So we think the lacto is actually playing an essential role to prime the land. And then if mom had the BIFs to pass on to baby, it would colonize better. So if there was no lacto, didn't pass through lacto, then maybe the BIFs from mom's gut, maybe it won't colonize as well. So it does sound like a little bit like all the stars have to align for baby's gut to be in the best shape, but that's how it seems like that's how nature intended it to be. However, because we have so many interventions today, modern interventions, like the process is being disturbed a little bit, which may explain why we see 1 in 2 kids today having at least one chronic condition.
Mhm.
And this is a stat from the CDC. So I do think we are living today in a pediatric chronic condition crisis,
Mhm.
where like if 1 in 2 kids have eczema or asthma, obesity, type 1 diabetes, that is the new norm, but that new norm should not be acceptable. That is just too high.
Absolutely.
We
Yeah,
have to make,
no, if you can start intervening at birth with the proper probiotics if necessary, that really could make a big difference for their long-term health.
100%.
So speaking of crises, I've heard numbers now for autism, like 1 in 26 kids or boys, I'm not sure which, is born, or is diagnosed with autism. And I'm curious, are you taking conditions when you take in these microbiome kits, and have you seen any kind of a microbiome signature associated with autism or with ADHD or anything like that?
Yeah, we haven't kind of done the work. Oh, well, we have done some work, but we haven't added that signature into our platform. We do have predictive signatures for microbiome signatures for eczema, asthma, uh, constipation, things like that, colic, but not yet autism and ADHD, just because the research there is quite new and we're still kind of untangling, because like for things like autism and ADHD, more likely than not is bidirectional. So is it the gut, like the imbalanced gut that's kind of causing the autism or behavioral issues, or is it the behavioral issues that's causing a bad gut? The answer is probably both of them, right? It kind of has a bidirectional effect, which makes it more complex and harder to untangle what we want to show. But there's definitely studies that show that if you if you modify the gut, it leads to behavioral issues. So there is definitely a gut-brain axis connection for sure.
Mhm.
And in some studies, I think because we do like more kid and infant studies, that we found a study where it has shown that an imbalanced gut in babies as young as 6 months old could be predictive of ADHD later in childhood. But again, that's just one paper.
Mhm.
So it's a lot to untangle, and our science team is very rigorous in the way that we want to see multiple papers supporting the same notion, the same taxa, the same microbial signatures before we put it forth.
Yeah.
But it's kind of emerging research, right? So we're continuing to track it. There's, we know that the gut bacteria produce metabolites, which can travel to the brain via the blood, so crossing the blood-brain barrier and influence brain functions and affecting mood, cognitive functions, and neurological health for both kids and adults. So we know there is a linkage. But yeah, we're in pretty early days on that work.
Okay. So I did the Tiny Health Gut Test, and mine was a pro test, but I don't know that I saw that offering on your website. Is that a new offering or something you have to go through a practitioner for? How does that work?
Yes, it is something that is practitioner only available, and I ordered that for you because you were probably familiar with the more conventional PCR tests like GI map, GI
Right,
flex.
right.
And so the pro tests that we offer has to be ordered by a licensed practitioner to help you interpret it. And the only difference between our regular test and the pro test is that it adds on the stoichemistry markers,
Right.
which are the calprotectin, secretory IgA, um, and all those markers. And
Yeah.
we do have it actually. If you go to tinyhealth.com, I think /store, if you scroll down a little bit, you'll see the pro test. If you click through it, it will bring you to the practitioner website. So we now do actually have a new partnership practitioner website called poweredbytiny.com. That's a bit more practitioner focused because we realized in the 2 years that we launched, a lot of patients or users are taking the results to their doctors. And that's how we have hundreds of doctors kind of in our network now because they want to learn more about how to interpret our tests. And a lot of them are functionally trained. They're integrative or holistic health practitioners, and they wanted stool chemistries. So we created the protests for them to have additional measurements of the host immune response and, things like that.
So does that mean that people can go to your website and order the protest and you provide the practitioner for that?
they have to order it through their
Okay,
practitioners.
they have to know the practitioner themselves. Okay.
Correct. Yeah, because they
Got
need someone to walk them through. I mean,
Okay.
in the future, we may work with, we do have our own health coaches. I think it's just a matter of, like, we walk folks through our shotgun sequencing results, the microbial part, because we are experts in it. In the stool chemistries, we feel like we need the right practitioners who know those markers really
Right.
well. So I think maybe in due time we will offer that. But right now they have to order it through their own practitioner.
Okay. So for the microbiome test, the metagenomic sequencing, right?
Yeah, that
Yeah.
was called shotgun metagenomic
Shotgun.
sequencing.
Yeah.
Mhm.
So you only asked for a tiny bit of stool, like the Q-tip wiping of it. So I'm just curious, how can you be sure that that properly and in proper proportions represents what's in the colon versus an entire vial of stool that are used for diagnostic tests like the GI map or the GI-FX?
I'm so glad you asked this question. We get asked this so much. And by the way, GI map and GI-FX are not diagnostic tests. They are qPCR based, but at best you can call them a screening test, but it's not diagnostic, because to really properly categorize it as diagnostic, you do have to go through an FDA approval, which there isn't such a thing right now in microbiome. Like the FDA doesn't have the proper regulation for the microbiome testing, stool testing, which is part of the kind of like criticism in a way in our industry, in the stool testing world. I mean, I wish there was more oversight, and in due time, I think they will come up with that. But for now, it's a screen. But anyway, the PCR tests, some companies have a culturing component, which I think is, not I think, but I know is less reliable. And for culturing, you do have to scoop, a bunch of stool. And if you're doing multiple cultures, basically the way we've used cultures for decades, right? Scientists would take a small amount of stool and put it in a petri dish and grow it under the lab conditions, right? So the reason why we don't use culture anymore is because, or like, at least we don't think it's reliable, is because it is biased towards what you're trying to look for. It it favors rapidly growing oxygen-loving microbes, whereas most of the microbes in the human gut are anaerobic, so oxygen intolerant bacteria. So that means, like, like E. coli is usually oxygen loving, so it grows more quickly in a petri dish than, say, Archimencia or bifidobacteria. So the problem with culture is that it's going to give you biased results that really is overblown. Clinically, I wouldn't trust any culture.
Yeah, I don't pay any attention to the culture if the test I'm looking at has it.
Yeah, so maybe some companies are still kind of using that and culturing some, and I wouldn't trust that at all. But so like when shotgun sequencing came up, is basically the next gen sequencing method that kind of sequences your entire gut. We sequence all the bacteria, viruses, parasites, fungi, or yeast, archaea, everything in your gut. Versus the way PCR tests work is that you need to know what you're probing for. You need primers for specific microbes that you're looking for. So typically in a GI map or GI-FX, you get 30 plus microbes you're looking for, but you may be missing the full picture of what's in your gut, right? So did the 35 microbes in that PCR test, does that represent maybe just 5% of your gut, or did that represent 95% of your gut? It makes a huge difference when you come to that level. So like, I think, PCR tests are great when you're looking for specific things, like if you have the presence of candida or not. But beyond that, it's kind of less useful for, helping you understand overall gut health, and do you have enough, fiber digestion functions, short-chain fatty acids, and things like that. But coming back to your question about then knowing technology, kind of like the way shotgun sequencing works is that it chops out all the bacteria and like all the microbes in your gut, and we reassemble the genomes to establish that you have 1% of E. coli strain D, 2% of Bifidobacteria infantis XYZ. There's been a few papers published that shows that changing with next gen sequencing, just taking a swab versus a stool sample yields the same results, and it represents your gut because the shotgun technology is able to chop up fragments, millions and millions of fragments in your gut. And it's pretty even. There has been tests showing that, kind of like sticking the swab into different parts of the stool versus just one part, we get the same results. Because again, that tiny little fragment of fecal matter, in a way, some labs call it like it's very well mixed. So
Okay.
it represents the
Good to know.
large intestine quite well. We know that because the scientists that work at Tiny Health, and they come from different universities, from Washington University or, Iowa and UCSF and things like that. Each lab usually does their own validation because even the scientists want to know if the swabs, how does that compare to the whole stool?
Mm-hmm.
So a lot of them are unpublished, and I wish these labs publish more of these papers, but I often hear them saying, we've done the test and it's so consistent that we switch from stool samples to swabs. And then also at Tiny Health, we have some families or some users just wanting to test how reliable and reproducible our tests are. They buy 2 swabs and sample it like one day apart. And oftentimes we come back because we have a very good QC process. We're like, this is the same person. Did they submit 2 swabs? And then so we email them to ask, because we would know it's the same person. Yeah, like it's twin, it's so identical. Maybe the top species, maybe the 5 top species might swap places just a little bit, but we know, I can tell looking at stool sample that this is your stool versus your gut versus someone else's gut.
Okay, great. Well, so I'm going to pull up my sample results and we can look at them together.
So we have this, you did the Pro Gut Health Test, which includes the stool chemistry markers, as we talked about. And this is like a good snapshot that overall your gut test looks pretty decent. You do have 7 things that need support, so I would focus on these areas. And then 6 things that can have optional support. So I would, in terms of priority, work on these first. And so the way we kind of categorize our tests is through different categories. Because again, I think sometimes stool tests focus too much on pathogenic or opportunistic pathogens and not enough on the balance of beneficial bacteria, protective bacteria. So we always start with do you even have those beneficial bacteria? And it seems like you are lacking some of them here. And then, and we also tell you what kind of probiotic species we detect in your gut, if we do detect them. And then do you have a lot of disruptive microbes that are opportunistic pathogens or parasites and infectious microbes and things like that?
No, let me just pause for a sec. I didn't make it totally clear. You can go to the show notes website to pull up this report so you can kind of look along with us if you're listening to the podcast.
Right. I'm just walking you through each category that we show. And so then the third category is gut inflammation markers. So this is what, maybe you might commonly hear of leaky gut. I would come here to see if you have a leaky gut. But again, based on these test results, you don't really have a leaky gut, which is nice. But what you're lacking here seems to be beneficial bacteria. And maybe some high levels, borderline high levels of pathogens here. No parasites, which is nice. Kind of going down, short chain fatty acids are essential for your gut lining. And the most important one for adults are butyrate. So you do want to see good butyrate levels. For kids and infants, acetate is the most important. So we do have, because you did the Pro test, we are measuring not just the microbial production capacity, do you have the microbial gene function to produce butyrate, acetate, and propionate? We also measured the actual concentration. So it seems like, in this results, your actual concentration, for the most part, is okay. It is a little bit low, and that's driven by your acetate being a little bit low. So that's kind of what it means. And the like microbial functions is not performing so well. So there are action items that you can take from a dietary perspective in your Tiny Health results portal. You're also going to see, like from a dietary perspective, what kinds of maybe specific fiber foods that will help with these metrics, and also supplements, right? So there may be, I know there are butyrate supplements. I
I
don't
sell one.
remember. You sell it? Oh, you do?
Yeah, I have a product
Amazing.
called Tributrin Max, which I take almost daily, but since I've changed my diet
Wow.
since these test results, I'm needing it less and less with all the fiber I'm getting.
Okay. Well, it seems your butyrate is okay. I'll need to dive into the butyrate producing capacity here. But yeah, it seems like acetate, and then I'm going to attempt to pronounce this, beta glucuronidase. I can never
Yeah.
pronounce it well.
That was the most alarming one that I saw.
Yeah, that one is, the high amounts, and you're not so high, you can kind of go down to the detail level on the bottom to see exactly how off you are. But we can come back to that. I think you're just borderline high, which is usually linked to estrogen dominance, right? If it's too high, it's typically linked to like, if you're premenopausal kind of like state, it may cause issues in women's hormonal health. So this one is very important for women's health, especially as we get into those older ages, to look out for. And then digestion and absorption markers seem to have more fiber digestion, it's complex sugar digestion issues. And we also have some dietary recommendations there on how to improve these markers, because it may mean that eating specific fiber, and we'll have to see what kind of fiber is triggered in you, that you may feel a little bit discomfort in eating that. So like, this is also a question we get asked a lot. It's like, oh, if my gut isn't adapted to digest these fibers and eating them causes me discomfort, then why should I eat them, right? But if you do want to, ideally the human gut can handle a large variety of foods. So if you eat certain foods and you don't feel good, it may mean that, it's kind of like chicken and egg. You may eat more of that to encourage more bugs that can digest that, then that enables you to eat a variety of foods in the future. So ultimately, that's what we'd like to work towards.
Mm.
We don't, like some other stool tests tell you not to eat certain foods and eliminate it from your diet entirely. We don't do that because we believe that sometimes eliminating certain food groups out of your diet may lower diversity and cause more issues. Again, it depends on what your conditions you have. You have some people are on a FODMAP diet for a good reason if they have certain digestive issues, right? But if you're a healthy gut, if you have a relatively and you don't have these conditions, then we would really encourage a large diversity of foods. And then, yeah, like some of these other balance and robustness, you don't really have an overabundance of 1 or 2 microbes. Some people have that, which makes diversity very low. And then we have a population chart here that compares you with everyone who's taken Tiny Health tests. So 20, at this point, maybe we're closer to 30,000 samples. So definitely there are things you can work on. And I think you mentioned that you have had some conditions, right? And you were bloated when you were taking this test.
Yeah, I mean I have autoimmune SIBO, so I always have that, and I have been letting myself get sort of out of hand in preparation for a stool test or a breath test by my doctor to get a hold of some antibiotics. So
The SIBO bacteria that it's connected to the methane
yeah,
producer
no, I don't
here.
have that one. Yeah,
CBI, you don't have it. But
that's not my
again,
problem.
that's not your problem. And the breath test and all that there's no good diagnostic test for SIBO right now, because it is trying to measure what's in the small intestines, right? And overgrowth of bacteria in the small intestines. So right now there is no known test to measure what's in your small intestines. And all the stool tests that you find in the market are only measuring your large intestines. So it is definitely, even the breath test, which is the gold standard, is not perfect. It's very sensitive. But certainly if you have the symptoms, then there could be other things that are triggering the symptoms, right? So if you go down to page 5, this is where you go down to the detailed level and all those things that were triggered like you're low in bifidobacteria, but you do have infantis. So this is your native strain, which is nice. So maybe with a little bit of HMO and like the right fiber diet, high fiber diet, and maybe some probiotic help, you can boost these amounts to a higher level. And Archimensea, at least you have some. So Archimensea is really important for your gut lining and metabolic health.
Yeah, I take a pendulum product, the
Yeah.
metabolic daily. So I have
Mhm.
done that in the past and I'm doing it now. And but I of course I paused for a couple of weeks to take the test, but
Mhm.
something's still there. So I was glad to see that.
So it's good because I have zero and I have not, it did colonize temporarily when I was taking pendulum too. And as soon as I stopped, it kind of disappeared. And this one, if you don't have any, it's harder to engraft. It's just really
Mhm.
hard. The
Right.
fact that you have some is a really good sign. So again, if you have some, I think continue on the pendulum and the polyphenol rich foods and maybe an HMO and checking again, because you don't want this to be too high too, right? Because Archimensea is kind of a bacteria that replaces the mucus of the gut lining. So if you have too much, it's kind of degrading the mucus too quickly, and it could be linked to, eczema and skin issues as well. So you don't want that too high. We also see it shooting really high as a like inflammatory event, like it's kind of your immune system may be reacting to some pro-inflammatory events. So, but it's very temporary. When we see spikes to like 14%, something happened and it comes back down.
Ah,
So
okay.
yeah, it's very mysterious. Archimensea is a very mysterious bug, but we know that it is quite important and you do want it in, a few percentage points would be just nice. And Faecalibacteria it's an anti-inflammatory marker. This is important. Again, through a lot of dietary actions, you can help improve this bacteria. But let me just instead of going one by one, because this is a lot to go through, are there any specific areas that you want to double click into? We see a little bit of C. diff here, which is why it's yellow. And this pops up with prior antibiotic use. So I would ask if you've had prior antibiotic exposure.
It's been a long time,
Long time.
but I take antimicrobials herbs on a relatively regular basis. As soon as the SIBO starts acting up, I'll dose myself with herbs again. So
Mm, mhm, mm. Yeah, so we see some, not a whole lot, 0.05%, just, would say keep an eye on it. And if you do need, I mean, this is where, if you do need antibiotics, I would be more careful because we know this tends to bloom with antibiotic. It's antibiotic resistant, right?
mhm.
Yeah.
Yeah. Well, I can take some of my other product, my serum bovine immunoglobulins with it. So
I love SBIs. I do that when
yeah.
I travel. I do that when I have high pathogenic bacteria, gut inflammation, signs of gut inflammation. That works wonders to bind these toxins. So maybe it was higher before and you've worked it down. Because
Maybe.
again, your gut doesn't look that bad. Like I think,
I was pretty
um,
pleased with what I saw overall, except for the high beta glucuronidase. That was what bothered me because I know that predisposes you to breast cancer and to colon cancer. So I made a quick turnabout in my diet and have reduced meat drastically and have kicked up fiber and beans and lentils drastically.
yeah, beans and lentils can really help. Again, looking at the range here on page 9, you're just barely above, right? So I wouldn't be too alarmed. It is borderline. I think what you're doing right now should help. Hopefully if you retest in a couple of months, I would say maybe retest in 3 months.
Yeah.
Hopefully that gets back into the green range. We've seen some people up into the right, and they have a lot of health issues, hormonal health issues. Um,
Yeah, I've always been a bit estrogen dominant, but I'm in menopause now, so I can't imagine there's a lot of estrogen circulating in my system other than that's coming from the patch I'm wearing.
Yeah, for sure. So it's good to keep it in check for sure at this age. We were wondering what the fiber digestion thing that was flagged, so resistant starch was flagged. And this, if you eat rice or potatoes, I would say cool it down first. I used to eat, as I'm Asian, oh, hot rice and hot potatoes, we would just eat it hot. But now knowing what we know about resistant starch, We now wait for rice to cool down, and even bananas, which I love bananas. I'm trying to buy more green bananas
Mm.
because they retain more resistant starch. So if you're low in that, in our action plan, we do offer some of these tips on how to increase that.
Yeah, no, I make these grape leaves, and part of them is rice and part is lentils on the inside, and then a dressing, and it's got oregano in it, and they are just like the perfect resistant starch food and fiber
Yum.
food, and I just am addicted to them.
Yeah, that's great. So yeah, more of that perhaps, or different variety of them, like maybe add some green bananas or like something to your smoothies. Yeah, and then this one you have 0 RPKM for. This one I am a little bit less familiar with, so I'll have to go back into the web portal to figure out IMOs.
Isomalto-oligosaccharides. Basically you're saying that I don't have the bacteria to digest those. Is that what it's essentially saying?
Correct. Not just the bacteria, but the gene function to digest these complex sugars. So I think maybe it's correlated to your low BIFs, perhaps. So increasing the BIFs should help with this.
Mhm.
And it can be found in fermented foods. Um, how much fermented foods do you
I
do?
make my own sauerkraut and I eat it every morning. I don't, that's pretty much it. I occasionally used to eat yogurts, but every time I ate it, I felt sick. If I eat the entire
Mm,
container, I feel sick. I feel nauseous. And I don't know if
interesting. Maybe it's
that's,
related to this.
yeah, maybe I'm missing what one needs, but
Mhm,
or maybe it's the strains of bacteria and they're starting to ferment in my stomach. I'm not sure what, but
mhm.
I'm
Yeah.
gonna try the Bifido yogurt with the Evivo and see if that helps things.
Yeah, yeah, and if possible, add an
Add
HMO
the
supplement.
HMOs, yeah.
Yeah, the Leo
I'll have to get
origin.
the HMOs too.
Yeah, and if you can tolerate, adding some sauerkraut, like bit by bit, or kimchi, and some of these fermented kefir, maybe like bit by bit, will help you hopefully
I,
gather some
yeah,
functions here.
I have been eating sourdough bread, but aren't they? Oh, okay. So these are not
Yeah, sometimes it's
bad.
like if you've been doing one thing over and over again and it's not showing the results, maybe it's, a lot of times it's the variety or the kinds of, maybe like
Yeah,
one thing is just not working and
yeah.
your gut needs a different thing. And oftentimes it's a variety of things, right?
Mhm.
So yeah, we could try that. Um, but let me see. Everything else, your vitamin production is perfect. Not everyone has all of this in green. So I would say this is very good. No
And that's all
hysterectomy.
pulling from the bacteria that I have or the amount of vitamins that are found in my stool.
Oh yeah, this is a good clarification. No, this is the again the gene function of the bacteria. So not the bacteria directly, but your bacteria has the capacity to produce these vitamins. And like if you have low amounts of them, it just may mean that you have to eat more of some certain foods that give you more of that kind of vitamin. But it doesn't measure the vitamins in your gut. It just tells you that the other essential role of your gut bacteria is to produce vitamins. Like I think this is a very like not a very well-known fact, but your bacteria gives you vitamins, which is amazing, right?
Yeah,
So it doesn't mean you're deficient. It just means that the contribution of your gut bacteria to this vitamin is either sufficient or lacking,
yeah,
if that makes sense. Yeah, and then you have to fill that more with dietary means. Like if
right.
you're low in B12, which is a lot of like people are often very low in B12, then you may need to supplement with the B12 um, supplement or eat more B12 rich foods, if that makes sense.
Oh, if you want to pause just a sec on the phyla, I
Mhm.
I was really glad to see that I had a normal level of proteobacteria, not an elevated level, because the last stool test that I did of this nature, which was a 16S, I think 50% of my microbiome was proteobacteria.
Oh,
And I have
wow.
since been supplementing with butyrate, which has clearly turned that whole picture around.
Yeah. Yeah. 16S is one of those earlier next-gen sequencing methods to like give you the full picture of your gut bacteria, which was a huge innovation. But I would say now, today, 16S is a very outdated technology. And because it can only detect bacteria, not fungi or parasites or viruses, it's also missing a lot of pictures. The way I would compare 16S with a shotgun is that you may take a photograph of your family members, and 16S gives you a blurry picture. So you don't know if that's uncle, like in an extended family, that's Uncle Tom or Uncle Tim, or Auntie Laura or Auntie Kim. Whereas shotgun sequencing gives you a very sharp, high resolution picture of who, and you can see Uncle Tom smiling and Uncle Tim like frowning. It gives you that specific
The strain.
picture. The strain, exactly what they're doing, what they're functioning, right? Are they happy? Are they not?
Yeah.
Whereas 16S may have high positive, false positives in that it may be mixing up, it may be confusing, an E. coli bacteria with something else that's not E. coli. And so you may see higher levels of something that, maybe is a little bit like, it's something different. So again, like maybe, I don't know, like 6 years ago, 8 years ago, that would be the technology, but we've really moved on from that since.
Okay.
Yeah. Yeah, so this is good. I think, it's again, a very stable DNA sequencing is very stable. If you were to do this stool test, like, a day apart, we'd probably get the same results.
Yeah.
It's very consistent.
And what's
So
Shannon diversity?
Shannon diversity is comparing the diversity of your microbes, right? So like the evenness meaning you don't have like really high overabundant bacteria on top, and then the rest are kind of like little. And you have a lot of, like I mentioned, our grown up adult microbiome has maybe 300 to 500 seats. So the more seats, the more species we find in the gut, the more robust and healthier your gut is. And you have a decent amount, it can be higher. So if we go down to your species breakdown, your microbiome breakdown here, we're on page 11, which is the last part of the PDF report. And if you go to the app, you can actually expand each species to look at them. And there are like citations, you can link out to PubMed articles to look at why we categorize these as variable bacteria. So you have 18% unknown bacteria, which just means that scientists haven't been able to categorize if these are good or friendly or unfriendly bacteria. It's just less studied. But we can see, like, I don't, I can't count, but if you were to count the number of species you have in the gut, because you're in the gray, you may have maybe closer to 300 than 500, right? If you were in the green, maybe you have 600 species, and the higher, the better, right?
Yeah.
So you kind of want to see, like typically when I look at a shotgun sequencing results, I want to see the top 10 species, what is really making up the bulk of your gut. And I want that to have more greens than yellow, right? Because then, again, like you have 12% of the top species, 10% and 4%, which is not bad. Like, what we don't want to see is this being 18% or 20%, which is like one bacteria dominating the gut. So the evenness, the Shannon diversity, the index is also linked to evenness. So if you have a higher Shannon diversity index, then this could be like more like 7, 7, or like 8, 7, 7, 6. It's more uniform throughout your gut, and like at least the first 5 to 10 species. So like, typically we want to ideally see some of these Glaucia, which is linked to, I think, butyrate function and things like that, being higher on top. And because you have your top 3 species as variable, like too high of these amounts of bacteria are linked to certain kind of conditions. Again, like they're not like terribly high right now, so I wouldn't be alarmed by these. But it's just very interesting to know, right? Like if you do have a family history of certain, like I see type 1 diabetes here and Crohn's disease here, Crohn's disease and type 1 diabetes. Type 1 diabetes again. So your top 3 species are kind of like, like has that again, not to freak you out on anything like that. But it's just something interesting that is connected to what's out there in literature, and we did detect it in your gut, something to look out for, perhaps.
Yeah. Fortunately, we don't have any of that in me or my family. So
Interesting. Yeah. So again, it may not be anything, right? Again, it's we see like when you see these conditions, it's connected to like higher levels in people who do have these conditions than other people who don't have the conditions. But
yeah.
like the DORI, for example, is 4%. It's pretty low. So I wouldn't be
Yeah.
concerned about that.
So we got to be wrapping this up. We've gone a
Mhm.
bit over time. So let me just
Yeah.
ask you, can people access their raw data when they do Tiny Health?
We don't automatically offer that, but we do have practitioners who write in asking for the CSV files if they're raw data to taxonomy files, and we manually produce them for people.
Okay. So it's possible, could a person do that for themselves or it's just a practitioner?
Yeah, I mean, anyone. I mean, I'm just curious why you, want that file. Like, are you running your own analysis?
Oh, some people like to upload those to websites like Biome Site. Some people track, uh, I guess if all the strains are listed in that report, which I assume they are, then that's good. Because
Mhm.
there was a couple strains I was worried about that are they're not good for your oral microbiome, and I had seen them in a prior metagenomic sequencing and have been using an oral rinse to try and clear them out. And
Mhm.
sure enough, they were not on this report. I was really happy to see that. This was the Porphyromonas gingivalis and the Fusobacterium nucleatum, which together the combo, puts you at risk of like pancreatic cancer and other stuff. So I was really kind of worried that I had
Oh,
had both
wow.
of those in the
Oh, yeah.
previous
You
report.
you don't want to see any oral bacteria in your gut, like for sure. It's
Yeah.
not a good sign usually. But yeah, we're glad we didn't see any of that. Yeah, we can definitely share the taxonomy files with you.
Okay. So I know that you offer like a membership option. Can you just tell people a little bit about what offerings you have now on the
Mhm.
site so they know, and then we'll wrap it up.
Yeah, for sure. Thank you. So we do have a membership program called Tiny Plus, where you do get 2 kits up front and a consult call with our microbiome specialist. It's a 30-minute call to walk you through your report. Um, the membership is $3.99. And I believe you may have a coupon code, right, to share with your audience. If not, we will get you one.
I know I got
Um,
a link, and it may be that the coupon applies on the link. I'll put it all in the show notes.
Okay, sounds great. Yes. So we did have that option because, again, we think that this shouldn't be a single time point. Whenever we have 2 data points of the same gut, we see the trajectory of change. And especially as it relates to early life, we want to see if it's trending and maturing in the right direction, right? And as for an adult gut or an older kid, it's proactively like, um, updating your baseline every 6 months is a good idea because you never know when you need medication, when a diet change, when you move or get sick with a stomach bug or need antibiotics. So then having that baseline is really helpful to help you recover and restore the missing bugs. So I would say, like, for those reasons, the membership is really good as a proactive step for your overall well-being and health. Especially if you have an infant, if you're trying to conceive and pregnant. I would even do 4 times a year to sample the infant. Um, we also have targeted programs, what we call targeted programs for people who are dealing with a chronic condition. So for kids, again, commonly it's a lot of these symptoms we talked about earlier, like eczema, allergies, constipation, even sleep issues. Older kids have different sets of issues, mostly constipation. And then, for women who are experiencing fertility issues, we do have a women's health program that could be a combination of a vaginal and a stool test, or just vaginal tests. So we have that too. For adults, we actually are coming up with a few digestive health sort of programs to help you tackle some of these conditions as well. But if all that, we also have a single, oh, sorry, my kid is back. A single kit where you can buy for $2.49 that comes with a consult call, and you can kind of like figure out if you want to commit to a membership after that or a targeted program.
Okay. And I did find the discount code is called the Perfect Stool for $20 off, all caps.
Perfect. Great.
Great. Anything else you would like to share with us before we sign off?
No, you can find us at tinyhealth.com. And if you're a practitioner, we do have a practitioner program to support you in consulting your clients. We train you for free, and you can find out about that through heartbytiny.com/practitioners. And then you can also reach me at hello@tinyhealth.com. I always do read a lot of the emails coming in, and on Instagram at tiny.health.
Okay.
Okay.
Awesome. Well, thank
All
you so
right.
much.
Thank you.